Welcome to the World Potato Congress webinar on potato common scab. Thank you for attending. Please submit questions in the question and answer tab, and we will address some questions at the end of the presentation. A recorded version of this webinar will be available at the World Potato Congress website at potatocongress.org under the Webinars tab. We value and appreciate our sustaining partners. I'll just take a moment to pause on this screen to acknowledge those partners. An informative introduction of myself. My name is Amanda Gevens. I'm a professor and extension plant pathologist with the University of Wisconsin-Madison and the University of Wisconsin Extension. I also have administrative appointments in our Department of Plant Pathology as associate chair and as the administrative director of our Wisconsin seed potato certification program. So I was asked to provide a presentation on potato common scab, and this disease is one of continuing concern globally in potato production. And so the topic today is biology and integrated management of potato common scab. Common scab is quite important, and in regions in which potatoes are grown, we have an appreciation for this disease. We don't always have an economic value to attach to the losses of quality, primarily quality that are attributed to common scab. But Canada does have a number that we can refer to, and that's $15 to $17 million in losses on an annual basis. This disease has substantial negative global impact, can reduce profitability primarily due to the quality in paraderm integrity as well as appearance for fresh market tubers. Typically, the scab does not impact total yields, but causes these blemishes and corky lesions. This can impact the eyes or the growth points of the tubers and can in fact affect the quality and health of the tubers in storage, as these lesions are broken or damaged paraderm that can be the sites for other secondary infections. And the severe scab can result in increased peeling losses in the processing industries. Common scab is caused by a number of species of the soil filamentous bacterium Streptomyces. This particular bacterium is gram-positive, and as we call it filamentous, this means that it grows a lot more like a fungus than it does like a bacterium, and we treat it in many ways like a fungus rather than a bacterium, but it functions mechanistically like a bacterium because that's what it is in the soil. Streptomyces will produce a phytotoxin called thaxtamin, which is what causes scab and pit symptoms on the potato tubers and other root crops. We can see this on carrot and table beet, which I'm showing here on the left-hand side of the slide. But primarily, this is a disease which causes greatest concern on potato. This thaxtamin is a pathogenicity factor, and what that means is that the production of thaxtamin is essential for disease to be created. And so we can have species and strains of Streptomyces species that are not pathogenic on potato if they do not produce thaxtamin. And so that is the key factor and a focus of many studies around the globe for enhancing control of this particular disease. Now, I want to discern here the difference between common scab and powdery scab. They're two very nasty diseases on potato, and scabs can look very similar. And so on the left-hand side of the screen, these are typical symptoms of common scab caused by Streptomyces, primarily scabies as a species, and that will be the focus of this particular webinar. On the right-hand side of this slide, you'll see pictures of symptoms of powdery scab. This is caused by a plasmodiophorid pathogen. This is a slime mold-like pathogen called Spongospora subterranea. And this particular pathogen creates tuber symptoms that are somewhat similar to those caused by the pathogen for making common scab, but will also create true root symptoms. which are these nodules on the roots that can affect growth and development of the plant. A bit further here, these are different in that common scab is typically favored by warm and dry soils. Powdery scab is typically favored by cool and wet soils. They do have unique cultural management strategies because of the differences in the pathogens, but both can be seed and soil borne, and both are extremely challenging to manage. And so understanding which particular pathogen you have is very important for the correct management of these diseases. But again, today the focus is on common scab. The disease cycle of the common scab pathogen in potato is one that is quite intractable in that the pathogen can be residing in the soil itself. And so even the planting of clean seed can result in common scab disease on tubers if the pathogen is present in the soil. But the pathogen can also be spread on the seed potatoes that may carry the pathogen in those lesions in the symptoms that were shown previously. And so we have these two potential sources of inoculum that introduce the pathogen into the crop. Ideal conditions for common scab include low soil moisture, typically less than about 65% during tuber initiation, soil pH between 5.2 and 8, and this can vary depending upon soil type, location, and the pathogen population. So we have different species of streptomycetes that have different requirements for pH. And daytime soil temperatures above 70 degrees Fahrenheit, which is just over 21 degrees Celsius. This thiaxamin, as I mentioned, is the phytotoxin essential to make disease. There are other elements which can moderate or regulate the intensity of symptoms or the virulence of this particular disease. And so what I'm showing here is a table that indicates on the left, there's a column called virulence value. And so this is often the way in which we will rate potato tubers for common scab when we're studying varieties, when we're studying treatment schemes to try to manage common scab. We'll use this value. And so 0 would mean no disease, 1 would mean superficial lesions or minor symptomology, 2 and then upwards of 3 would be more intense symptoms that may be pitted. And so when we look at this scheme in this table, that thiaxamin, this is a gene which controls the production of thiaxamin or the phytotoxin, which makes disease. It's in this case, we were looking at thiaxamin AB. So if that gene is present, we will have disease. We will have common scab. So that gene has to be present in the pathogen to make disease. When we look at these other accessory genes, or what we call virulence factors, we see here NEC1 and TOM-A. They're just two that we've looked at in my laboratory in Wisconsin. With the presence of these additional virulence factors, we see sort of a turning up of or an amplifying of symptoms in potato tubers. So if we have all of these present, the thiaxamin, the NEC1, and the TOM-A in this case, we were seeing the most aggressive symptomology on potato tubers. So here's just a look at the symptoms that we were able to generate using potato common scab isolates that had those unique profiles of thiaxamin genes and the virulence genes. On the far left, this picture is showing the netted lesion phenotype, which was correlated with the presence of the thiaxamin gene, but we had no other virulence genes in that particular isolate of the pathogen. So just the net scab. When we look at the picture in the center, these are potato tubers that were inoculated with a strain type of the common scab pathogen that had the thiaxamin gene as well as one of the virulence genes, in this case, just the TOM-A. And then the picture on the right is showing the most severe symptomology. And here's where we had the presence of the thiaxamin gene as well as TOM-A and the NEC1 virulence genes, which gave us the most aggressive level of infection. And we were seeing the pit scab. And so I show this just to indicate that there certainly are variations of common scab symptoms. on potato, and this is likely caused, this is in our evidenced by our laboratory and others around the globe, that that it has to do with the genetics in the particular Streptomyces pathogen that gives us these differential symptoms. Certainly, variety can play a role in this. Environmental conditions can play a role in this. In our study here, we were looking at soil that had previously been heat killed or heat sterilized, and then we were adding in the pathogen of interest with susceptible clean potato seed. And so in this case, this is the outcome of these three unique strain types of Streptomyces scabies. And when we think about which species of Streptomyces make common scab, there are many. And in having a look at studies evaluating the populations of Streptomyces affecting potato over the last 20 years, we find about a dozen different species of Streptomyces that can cause disease. And this can vary based on region. It can vary based on soil type, including pH of that soil. And so here's a listing. This is just something I generated from the internet to search on all the studies that were available. And Streptomyces scabies is certainly the top and the most predominant species type that cause common scab. But we also see acidoscabies, turgidoscabies, europascabii, stelloscabii, and the list continues. These different species have different attributes. They can make symptoms that may look a little bit different, and they can be active as pathogens under slightly different soil and environmental conditions. Here in Wisconsin, we conducted a survey of Streptomyces species affecting potato 10 years back. We did this in partnership with a researcher that works along the East Coast of the United States, or did formerly, named Dr. Leslie Warner. And Leslie had worked in our state back in the early 2000s, and so had surveyed Streptomyces affecting potato at that time with my predecessor, Dr. Walter Stevenson. And so we were able to have a look at differences in these pathogenic Streptomyces populations over the course of about 10 years. And what we found back in 2015 is that we saw consistently a predominance of Streptomyces scabies. So in looking at this figure, what you see on the left on that y-axis are a number of counties or just geographical locations within the state of Wisconsin. And those green bars, sections of the bars, indicate Streptomyces scabies as the species. When we look at the other colorations on the bar, these represent other species. So the orange represents Streptomyces stelloscabii, and the pink represents Sauropascabii. And we've seen a number of other species in our state as well, but the predominant type affecting potato is Streptomyces scabies. These unique species are pathogenic in a number of ways. And so one of the facets of pathogenicity that we looked at was the production of thaxtamin, the phytotoxin, which creates the symptoms. And so it's one capacity alone that just the strain type will make thaxtamin and make disease, but it's another facet that it may make it in greater abundance. And so the more thaxtamin you have is typically aligned with the most aggressive common scab symptom on potato tubers. And these can be attributes of unique species. And so when we looked at thaxtamin production in Streptomyces scabies on 20 strains that we collected from our state, we saw the most abundant quantity of thaxtamin. And this thaxtamin can be measured directly from cultures of the pathogen. What we had done in our program was to first screen all of the Streptomyces we collected off of common scab symptomatic potato tubers. And then we tested them for pathogenicity on radish seedlings, which is what you're seeing in the photos on the left. And if the pathogen did not grow at all, we did not find them to be pathogens of potato, but if they did grow and they limited the development of those radish seedlings, that indicated pathogenicity. And then from there, we progressed with an analysis of quantification of the Thaxton phytotoxin to characterize our populations a bit more so. And so as we look down this table, we see Streptomyces stelloscabii making also quite an abundance of Thaxtamin, as well as Streptomyces botropensis. We see another species, Europascabii, also producing quite a bit of Thaxtamin. And then some of the other species here that we see that are typically associated with net scab have a bit less Thaxtamin. So just a review back to this slide again, the development of Thaxtamin and the associated virulence genes seem to influence the common scab symptomology on potatoes. And so we start looking into management schemes, and understanding the way the pathogen works is critical to better managing this particular pathogen. And so one approach that we've looked after, as have a number of other research programs around the globe, is to see if we can interfere with that Thaxtamin activity. That Thaxtamin is essential to make disease, but it can be disrupted with auxin induction. And so this was discovered over time through the use of 2,4-D in potato. In some locations, this particular herbicide is used in low dose for enhancing the reddening or red skin attribute of tubers. And so in that process, it was also discovered that a reduction in common scab was seen. And so folks have explored this auxin induction in potato to see if we can manage it better to reduce common scab. What is found is that there's not a direct interaction of auxin induction and the pathogen itself. So when you add an auxin to a culture of Streptomyces scabies, for example, it does not limit the growth of the pathogen. But if you induce auxin within the plant, it makes the plant more resistant to symptom development in the common scab cycle. And so we looked at this in the state of Wisconsin for a couple of years. I'm just showing just one slide here from 1 year of data out of our North Central common scab study from back in 2019. We've looked at this in a number of additional years as well, but we looked at auxin inducers and we compared them to conventional treatments for common scab here in our region. And that was, in our case, blocker or pentachloronitrobenzene, which I have here listed as PCNB. We looked at two auxins. We looked at Hortus, which was indole-3-butyric acid rooting hormone, and we looked at Rejuvenate, which was 1-naphthalene acetic acid and potassium salt. What we found is that if we added the greater the number of amendments of auxin that we added, we did see a greater effect of some reduction in common scab, but it wasn't significant. And so the data that I'm sharing here was a year in which we did see numerically reduction in common scab severity with use of the Hortus as an in-furrow treatment and applied twice on the foliage of the plant. And so we induced this auxin within the plant, and we did see a reduction in common scab severity by about 4%. This reduction in incidence of common scab was at about 5%. And so those differences there, when we speak of common scab incidence, it has to do simply with the presence of a common scab symptom on the tubers at the time of harvest. And then the severity refers to the magnitude of disease. So severity would, it suggests the percentage of coverage of the potato tuber with common scab lesions. And so what we find in time and repeated years of work like this is that we would see some years with an advantage of applying additional auxin inducers to reduce common scab, but in other years, we had no effect with this treatment. And so again, while numerically it looked like it was doing something more, in subsequent years, we could not maintain this level of control in time. We've also looked at cut versus whole seed potatoes, and we've looked at the percentage of disease on seed potatoes during planting. And what we have found in our work is that whether the seed potatoes had 5% severity of common scab on seed or 50% severity, we had a very similar outcome of percent severity of common scab on the resulting daughter tubers. And so this tells us that even a small amount of common scab on your seed can contribute to a substantial level of infection on those daughter tubers. And so really it's best to have as close to zero as possible. And I know in some regions this can be a challenge to achieve, but the smaller the severity of common scab, the better for the health of those daughter tubers. When we looked at cut versus whole seed potatoes, and this was a question that came to us from farmers that were noting that when they used whole seed potatoes, they would at times see greater common scab levels on the daughter tubers compared to cut seed. And so we looked into this. And aside from the agronomic differences that we do see resulting from cut versus whole seed, we have a difference in timing of emergence. But we did see a slight difference. There was a trend of reduced common scab with the cut seed when compared to the whole seed. And there we think there may have been a difference in the vigor of the plant. But we also noted that it also meant a reduction in inoculum, because when we were cutting that seed piece into a 2-ounce seed piece, there's less skin, right? Less periderm, and presumably less inoculum going in with that seed piece than when we planted a whole seed tuber that had a greater area of periderm and potential inoculum on that seed piece. So managing common scab, overall, the resistance in cultivars is not well understood, but that is foundationally the place in which we need more research, right? We understand that it's probably multigenic and it's a complicated interaction of the skin with that thaxtamen of the pathogens, but we are still only scratching the surface of our understanding in the peripheral factors that influence common scab. And so the genetics of host resistance are always in need of further exploration. But that is foundational. When we consider disease management, always the best and most durable management form is in resistance from our host. And so moving forward, we look at other both cultural as well as chemical inputs to try to manage this very concerning disease. Over time, scientists have looked at disease suppressive rotations with green manures using buckwheat, canola, oat, rye, or millet. These can all inhibit streptomyces. The management of soil moisture, soil pH, and manure incorporation, if used, can all help limit common scab. And when it comes to the chemical and biological or biopesticidal treatments, some have greater repeat effect, meaning they're more reliable in effect in reducing common scab, but others tend to be more inconsistent in effectiveness. And that's where our understanding becomes less thorough. And so what we'll explore here in the remaining slides is management, including biological and biopesticides, conventional inputs. We will also look at fumigants and other elemental inputs and how they've resulted in management over approximately a decade of exploration here in Wisconsin and beyond. So as we enter into this section of treatments and their effects, I just wanted to remind us of the many factors that play into common scab development and severity. In a soil study that I collaborated on with my colleague, Dr. Richard Langkow, who is a soil ecologist with our Department of Plant Pathology, what we found is that there are many factors which certainly can influence common scab severity, including other microbial components, the bacterial populations, the fungal populations within the soil, as well as soil chemistry, management inputs. But of all of these factors, The one that was most tightly related to common scab severity was potato variety. So again, back to that one of the previous slides where I mentioned varietal resistance as really the foundational piece for most durable resistance. What was also what we determined to be the case with our soil testing and common scab studies in Wisconsin. All right, so soil pH, it can have influence. I think this is always a question of how low do we go? Some regions will refer to 5.0 or 5.2 as a target for reducing common scab. Other regions may look at 5.5 as a threshold for helping to reduce common scab. And this is very dependent upon soil type, and it is dependent upon the species and the strains of the species of streptomycetes that are predominant in your growing region. It's not controlled, doesn't guarantee control of common scab if you have a low pH, but it is one modifier of risk, which we know can help, combined with moisture control, rotation, resistant varieties, and other cultural practices. So just want to remind us of that as we move forward in thinking about management. And then finally, the soil type. The physical and biotic properties of each soil type can interact with the common scab pathogen and influence disease. Again, I'm listing some of these factors we've already discussed, but I did want to just reiterate these points as we enter into some efficacy data that I will share from just one growing region in north central Wisconsin. And so the results that we find may not be consistent with findings in other places of the world, given the unique pathogen populations and physical and biotic properties of the soil. All right, so in looking at fumigations here in north central Wisconsin over, I'm sharing in total 8 or 9 years of trial data, but we did not look at every input in all of those years. But I just wanted to summarize some of our work. Overall, our work in looking at fumigants resulted in good outcomes of reduced common scab and high yield. When we looked at these chlorpicrin inputs, some of them with biological biopesticides, as well as plant extracts like regalia, did give us very good effectiveness in reducing common scab and gave us high yielding crops. In Wisconsin, when I started 16 years ago, I did learn that some growers were concerned that the conventional use of metam sodium or VAPAM as a soil fumigant prior to planting of potatoes could actually make common scab worse. And so we explored this for a number of years. We've not found that to be the case. We did actually see some reduction of common scab, but it was variable between years. And this fumigant is used primarily in Wisconsin for the reduction of potato early dying caused by verticillium and root lesion nematode. And so in just looking at this listing here, by and large, all of those that contain chlorpicrin gave us good yield and good common scab control and high yield. When we looked at conventional fungicides, again, while fungicides are targeting fungi and not specifically soil bacteria, they can also play a role in common scab management. Some of this is for reasons we don't fully understand, but we do know that management of other diseases can help a plant maintain its good health, and a healthier plant is one that can resist disease infection. And so I think that is in some cases the mechanisms by which these fungicides are working to help us reduce common scab. But it may also be the case that there's an interaction with the chemistry and the pathogen that we don't fully understand. And so here I have listed some of the treatments that worked what I would term good. And those included blocker, as I've mentioned earlier, this PCNB, that is a conventional standard here in our region for common scab control. We did in a couple of years mix this with MoCap. This is ethoprop or an older insecticide. This work was done because there were some lingering questions about the role of insect pests in actually eating the common scab lesions that were pitted, and it was thought for a while that there was a role for insects in actually creating those pits. And so folks were looking at this ethoprobe for common scab control probably 20 years back. And we picked up this work to just further demonstrate that it wasn't, in this case, we did see some good control with MoCap alone in reducing common scab, but it wasn't because of that insecticidal piece. There is some other interaction which seems to be helping to reduce common scab symptomology with use of that ethoprobe, and we don't fully understand it. We discontinued our evaluations of MoCap or ethoprobe after about 3 years of work, and focused on other fungicides and inputs moving forward that had some questions and had some interesting interactions. This listing also shows you the rejuvenate and the Hortus or the auxin inducers that I mentioned earlier in the presentation. And again, some of these have had some good effects, but with variability. I had listed Quadris here, and while not a fungicide that we typically consider for common scab control, we had looked at it because it is a grower standard when applied in furrow for Rhizoctonia control and other fungal pathogens here in Wisconsin. And so we put it in to have a look at its effect on common scab. We found it to be highly variable. But in some years, we did see an exacerbation or a worsening of common scab with use of Quadris in furrow in this one location. And so I do want to mention this. It was something we saw at times in our field trial in the Langlade silt loam soil type that we were working within in these years. It is not seen around the rest of our state in other soil types, and it's not a condition that I routinely hear about from other growing regions, but I do want to make mention of it here. The other treatments that we've not looked at yet, but others around the globe have, include fludeoxonil. And so this is a common fungicide in the seed treatment Maxim, for example. And fludeoxonil is also being used in as a premix in some foliar fungicides now as well. But this fludeoxonil as a seed treatment has demonstrated reduction of common scab between about 25 to 60% in some field trials over recent years. And so, excuse me, this is an area in which we'll be doing some further work here in Wisconsin to better understand. But I wanted to make mention of this, even though it's not my work out of my program. It's great work out of others' programs, and it just shows us we have more to learn about the effectiveness of these fungicides, which are currently registered for other disease targets. But if they work for common scab, we could certainly benefit from improved understanding of that role. All right, and the biological and biopesticide evaluations were also conducted. Here I've listed some of the true biologicals that contain microbes, and they're functioning as a fungicide as microbes. Others are functioning because they're an extracted element from a plant or from a microbe, and it's within that extract that we have activity. And as we see in this listing here, most generally we see poor or variable effects of most of these biologicals and biopesticides. In some cases, an explanation is in the form of the variability, right, of whether an environmental system on a microbe that requires certain conditions to thrive, right? And so we have an expectation that a microbe is functioning, and that can be inconsistent given the environmental conditions in which it's growing. And so that is something to keep in mind when using microbes that are we expect them to live and thrive and grow in order to do their work. if the conditions aren't right for them to do that, they won't be as effective, right? And in general, biopesticides and biologicals tend to do better as fungicides or disease control agents when the disease pressure is low. The one exception I'll mention, when we have looked at Bacillus amyloliquefactions treatments over time, and this would include double nickel, Amy Protec, and there are a number of others, different names that these products are registered under across the globe. But these have been some of the most consistent treatments as in-furrow and seed treatments for the control of common scab. And so I'll just point that out in the lower portion of this table. We've listed these Bacillus amyloliquefactions treatments, and the comment there on general efficacy is that they've been good, right? These are good. Some are variable, but by and large they're good for common scab control in most years that we have studied them. All right, and in summarizing here, we continue to look at a multifaceted approach to manage common scab, and it's critical for farms to consider all aspects. So that's irrigation management during tuber formation. That's modification of soil pH to try to limit the success of the common scab pathogens. Crop rotational strategies to help reduce that pathogen population as possible. Biological controls. These can work under certain controlled conditions when the environment is right for promotion of the biological, but they can be variable. Disease-free seed tubers is important, as we know they play a role in introducing inoculum to the field. Chemical fumigation and seed treatments or in-furrow treatments with fungicides can all be part of a management strategy to help reduce common scab on tubers. Sulfur fertilizer, this can help reduce common scab severity, whether that's through reduction in pH or as a direct fungicide on the pathogen. And ultimately, use of resistant potato cultivars when possible. This is certainly the most desirable approach and most reliable method. All right, and I wanted to just end with a couple of informational slides. There's a lot here, and I'm not going to read off all of these listings, but I just wanted to make mention that if you have interest in better understanding the streptomycete population that is affecting potato in your region, there are laboratories around the world that can help get you the information you need. And so the caveat here that the labs that offer these types of services, they may need to meet with you and have a consultation to understand what you need. So there is certainly a detection of species that create disease on potatoes. So they would have to do some, whether that's a PCR work or a culturing and pathogen ID. But it is possible for a lab to help you determine what species you have and to help you understand if the species of streptomycetes are in fact producing thaxtamin and creating disease. And so there are a number of labs that offer variable services in helping with common scab understanding around the globe. When we consider the North American and the Asian laboratories, here again, there are about a dozen that came up in again, just trying to look briefly at labs that could offer this service. I know in my own laboratory, it takes us some lead time in order to do this work because we're not set up to do it on demand all the time. And so in understanding the streptomycetes of our soil, we have to have the right materials in the lab on hand in order to most effectively serve growers' needs, whether that's from potato tuber tissues or from soil. And so it is best to connect with the diagnosticians and the scientists doing the work before submitting soil or even undertaking a sampling, because they may have very specific protocols for you to sample. And so I'll just share this with you because I think more knowledge gives us better capacity to manage disease. And it's important to know what resources you have in your region to do just that. So with that, I thank you for your attention. And I think we can move to questions. Okay, I see one question here. If you have common scab in your seed, but you need that seed, what's the best route to minimize infection in the field? Yeah, that's a great question. And I think some of the practices mentioned at the end when we look at seed treatments can help reduce the effect of common scab in the developing daughter tubers. Certainly, management of soil moisture can help within that season, but um, the the treatment, again, most reliably in our region was blocker or PCNB at time of planting. And again, depending upon the pathogen population in your region, it may be that the Bacillus aeruginosa treatments can also assist in this, in reducing effect. And again, I don't know if others have further explored the auxin induction, but that, when applied as a foliar treatment, also resulted in a reduction in common scab severity in our hands. The question: is there any effect of scab on skin color of potato? That's an interesting question. I would think with netted scab, certainly there can be a dulling of the tuber, and I have seen purples and reds that with too much net scab you do get a dull or rough finish, almost kind of a russeted finish, and that can affect your skin color if that's where the question is coming from. But I think it can, depending on the scab type that you have, it can certainly affect the skin quality and color. There's a question about the soil analysis through which you can determine what kind of streptomyces you have, and yes, this would be done within laboratories to get at the heart of what species you have affecting common scab, as well as the attributes of those strains of the species. There's not a simple test for that, and so it's exploratory work, and it would require some consultation with those doing the diagnosis for you. What stage of tuber is most vulnerable to common scab? Typically, it's early on, and so common scab will infect at tuberization and further development of the tuber. So that's the most vulnerable stage. Typically, that's aligned with flowering and beyond. There's a question about the application of acidic pre-planting and or fertilizer materials such as ammonium sulfate. The question is, could this reliably reduce levels of common scab? Here again, the sulfurs can do two things, right? They can reduce pH. It's hard to reduce pH within a single growing season with treatments like this. We've tried to do some work in soils, but it's often not effective to try to reduce pH in season. But within that tuber sphere alone, you can certainly try to make that reduction meaningful if you're close to 5.5 or lower. But you can't make a gross jump from, say, a 7 to a 5.5 within a season, but you can probably reduce in smaller points. But the sulfur will also have a direct negative impact on the Streptomyces scabies pathogen, and so you're getting two approaches at reducing common scab with a treatment like sulfur. There's a question about using minuet or concentrated serenade. Um, we've looked at serenade soil and some other versions of Bacillus subtilis on common scab over time. We've seen less control with common scab with Bacillus subtilis than we have with Bacillus amyloliquefactions. And so that just my comment there, it does seem like there's a bit more activity with that Bacillus species, amyloliquefactions versus subtilis, which is the serenade. Have we looked at Elatus or Amesto for common scab control? Indirectly, I have done this. I need to look at my data and go back over time. So Elatus, of course, is solatenol and azoxystrobin as an in-furrow, which has demonstrated in good control of diseases like rhizoctonia and silver scurf and some black dot. Amesto has two active ingredients, and that's a seed treatment as well. And they're the FRAC7s, right, or the SDHIs. I don't know that they have direct common scab control, but I haven't looked at this directly, and so it's a great question from Danny Johns. I know I myself am going to do some more work and look into that further, but not that I'm aware of. But we'll see once we get a hold of the numbers. It was a question about reducing pH. If your common scab is attacking between 5.2 and 8, then what should we use for getting that pH low from 8 to below 5.2? And at what pH can a potato plant no longer grow, right? If we get that too low. And those are all very, very good points, right? And so, right, when you get less than about 5.2, the plant is impacted, right? It is not, it's not accessing nutrients as well. And we know that's the case with other cropping crop plants as well, or rotational crops. And so getting that pH up for rotational systems is often important. But right, getting it down 3 whole units can be a challenge. And so, again, in some of the work that I've done in field trials, we have used sulfur to try to do this. And we couldn't really move the mark less than about a half or 0.5 pH in within one season with treatment. And I don't have the rates fresh in mind to share here, but it is I think it's unreasonable to expect a whole 3-point shift in pH within one season. So balancing this, given the your rotational cropping schemes, would be important. One question about how often are we seeing that combination of Thaxtamin AB, NEC1, and TOM-A in strains of Streptomyces species? And that's a great question. We looked at about 60 isolates or strains in our state, and probably roughly half to a third had all 3 of these. And so would have been some of the more aggressive strains to make some of the worst common scab. The balance of that 60 had Thaxtamin AB plus just one of those virulence genes. Very few of them had none, right? So where we just saw Thaxtamin, that would have been a weaker symptom or just a net scab. We had a smaller percentage of those in our total population of screened isolates in our study. There's some question about information on common scab control in mini tuber production systems such as aeroponics. It's a great question. I don't have anything specific to that system, but I would think, again, if you're starting from a microtuber or a tissue culture plantlet, you should not have pathogen present there. And so just thinking about what you're starting with, right? You shouldn't have inoculum on your starting tissue. And you wouldn't be introducing inoculum in a mist system. And so the risk should be very low, if not removed from that system entirely. So I guess I'm having a hard time thinking of where that introduction of the pathogen might come from, unless it is on the starting stock. But yeah, without the soil or presence of the pathogen on the seed, there wouldn't be another way for it to be introduced. And so it should be a clean system. It's a good question. Any work done on the addition of calcium in furrow to increase calcium concentration in the tuber, which maybe it makes it more. I would suppose the question is sort of suggestive that you have a more durable, healthier skin or periderm that might resist infection. It's a great question. I don't have an immediate answer to that, but I think that's a valuable question. The sulfur that we've used in our studies, we were using Tiger Sol. Um, and I'd have to look up the details of that. But it is Tiger, Tigersul, right, Tiger Sulf. Um, that's what we've used in our studies to date. I have not explored other formulations to share more beyond that. There's a question about whether the common scab pathogen can infect potato tubers at all stages or stations of tuber development. Um, and typically the infection is occurring early on in that initial tuber development. And so as it moves later, we I think what's hard to see or tell the difference between is if there's ongoing infection or if the symptoms on the tuber are just they're worsening as that tuber grows, because you will get some cracking and a sort of a change in the appearance of a symptom as the tuber ages and expands. And so can an infection continue? Usually not once the periderm is maturing. And so it tends to be an early tuber development infection that then worsens visibly in time because of that expansion of the lesion through enhanced cracking or more uh, corkiness as that tuber is responding to the thaxtamen over time. So question about a um, nature of the scab lesion. A symptom called fissure scab has been noted in South Africa associated with certain species. It looks a lot like growth cracks, but it is not. Have I noticed this in Wisconsin? I've not. It's a very interesting question. It's something I am thinking back to where I have seen cracks, and we don't always test the for common scab in those cases if we thought it was something else, right? And cracks can certainly be growth rate response. It could be herbicide response, and so I learned something here, and will I'll be mindful of that, Rick Peters. Thank you. Some questions about trichoderma or bioderma containing fertilizer applications in the field. Can that reduce affected or reduce common scab? I don't know of trichoderma significantly reducing common scab. And again, among the biologicals, I haven't seen trichoderma as one that reduces scab, but again, in with a fertilizer application, I'm not familiar with that particular input scheme to speak further on that. Does scab play a role in tuber rotting incidents and storage? It can. Certainly particularly pit scabs where you have damaged periderm, you have periderm that's not regulating that the tuber in terms of its respiration or metabolism, and so it's a tuber that it will be harder to manage in storage. But it's also a tuber that has, in essence, wounds on it. And so those wounds become the site for secondary and sometimes tertiary infections by other pathogens. And it could be Fusarium, it could be Pythium, and could be bacterial soft rot that enters into those wound sites, creating tuber rot. So certainly excessive common scab can play a role in further tuber rot. Is there a way to balance powdery scab and common scab management? That's a terrific question. Particularly given I always feel like they're quite opposing in that for common scab, you want some soil moisture, and that pathogen it's favored when the soil is dry and when we have warmer temps, whereas powdery scab is favored when you have wetter soils and cooler temps. And so they seem quite opposed in trying to manage these, but uh, yeah, to balance this, I think it is very much a challenge. Um, and we're still trying to better understand, I think, our management of powdery scab and even where it's present in fields still. Um, in our state, we've seen over the last 20 years an increase in the number of fields with powdery scab in the soil, and sort of how that is presenting in a field. And so typically it's in it's in lower areas where moisture collects more readily. So there's an increased understanding of this, but the field presence of powdery scab can be a bit different than common scab. Yeah, I think the question is spot on. How do we balance this when those conditions are quite opposed? Um, if possible, using cultivars with resistance. I know we have, I think our understanding of host resistance to both of these scabs is it's general based on I think field responses, but we don't have I think as great of a knowledge base in our powdery scab resistance as we do with common scab. But use of resistance I think would be critical if you can control one of these through your host resistance and then try to target the other with optimum practices would be maybe the way I would think about this. And there's someone here from the UK, David Nelson, who mentions irrigation and maintaining a moist soil during the 3 weeks immediately following tuber initiation provides very good scab control in the UK. We have looked at this as well. And again, it very dependent upon soil type and I think the populations of streptomycetes in the region. But certainly that is, it seems like from your experience, David, that soil moisture has a very pronounced effect on management of common scab, and that's really interesting to hear and great to hear that it's working. I think in some locations where maybe we have the likelihood of precipitation may be less, or access to irrigation may be limited in some ways. It's not a factor we can control as well, but yeah, quite dependent on the pathogen population, I think, in host resistance. It's a question about fluazinam, which is omega, had good control in the Wisconsin trials for common scab. Are these soil applications or foliar or combination? In our work, those were in furrow applications. So we applied the fluazinam at time of planting. As per our label is was for powdery scab, for other diseases as well, but that's how we applied the fluazinam in our common scab studies in Wisconsin. Okay, we're getting close to the end of our hour here. Just looking through some of these, I think I've addressed with previous questions, but I'm also quite happy to collect these questions, and if there's a way to interact with you directly via email, happy to follow up with anyone that has some lingering questions. They're great questions, and I think we have remaining much work to do in continuing to understand common scab management in potato here across the globe. So I thank you for your attention, and I thank you for your really thoughtful questions in this Q&A session.